That was the aha moment. Oh my God, like what is this, right? We didn't even know what a microbiome was. So when NIH came up with the Human Microbiome Project around 2006 or 7, lung was not a target organ. Lung has bacteria, but it's very low biomass. But low biomass doesn't mean that it's not important, right? Could be low in number, but could be modulating a lot of things. That's what we found in our clinical studies, that if there was specific kind of bacteria in the lungs, these infants born with that, they were more likely to either develop a disease or be resilient from it.
Hello and welcome back to the Smarter Not Harder podcast, your home for 1 cent solutions to $64,000 questions. I'm your host again today. My name is Dr. Scott Sherr, and it's a pleasure to be back with all of you. Today's podcast was a super cool one on the gut-lung axis. Have you heard of the gut-lung axis? Well, you will be soon, because there's some amazing therapeutics that are happening both directly into the airway, so intrapulmonary, as well as into the gut as well. And so I had a really fantastic individual, Dr. Vivek Lal, on the podcast today. And he's going to be one of our speakers at the Health Optimization Medicine and Practice Symposium, October 17th and 18th, here just outside of my hometown in Boulder, Colorado. So come check him out, learn all about the gut-lung axis and more. And here's a little bit of a bio on Dr. Lal, just to give you a feel for kind of the breadth of his work. He's a physician scientist and innovator based in Birmingham, Alabama. He is double board certified, having completed medical school, residency training, and clinical fellowship in neonatal and perinatal medicine. He completed a research fellowship in pulmonary and vascular biology at UT Southwestern, and an executive program in drug and device development from MIT in Boston. He serves as the director of clinical innovation at the Marnix Hersink Institute of Biomedical Innovation at the University of Alabama in Birmingham, and is director of Pulmonary Microbiome Lab. He's a serial entrepreneur. He's founder of BioStack Ventures, through which he launched successful companies such as ResBiotic Nutrition, which we talk about today, which makes consumer probiotics, Alveolus Bio, which is FDA-approved pulmonary drug development, and we talked about that too. He co-founded and serves on the board of Urgent Care for Children, a Southeast U.S.-based urgent care chain, and he's also the CEO of ResBiotic Nutrition. Vivek is a really cool guy. That's the short story. We had a really great conversation, really going through a lot of the questions I had about gut-lung axis. I didn't know what the gut-lung axis was, really. I mean, we talked about how they used to think that the lungs themselves were sterile, just like we used to think a lot of places in the body were sterile, but the lungs are not sterile. They have a core amount of bacteria that live in there. And so there are various ways to modulate the health of the lungs by what types of bacteria are in the lungs, and also what types of bacteria are in the gut, and how various types of metabolites from bacteria in the gut can help to enhance the inflammatory cascades that are occurring in the lungs in things like COPD, inflammatory or idiopathic pulmonary fibrosis, and other things like asthma, et cetera. So what Vivek has really been able to develop are therapeutics that go into the lungs directly, and also in the gut microbiome, various strains of lactobacillus specifically that can help repopulate the good types of bacteria in the lungs and the gut. There's so many potential different applications for this, which I'm super excited about. Things for like infection or colonization, many issues that we see in acute conventional pulmonary practice or in internal medicine practice, as I have had in the past, working as a hospitalist for many years, but also in the performance world, how you can see increases in FEV1, for example, by using a pulmonary focused probiotic that goes in the gut directly into the lungs itself as a mist. I love that Vivek said was air is medicine. He talks about that. And anybody that breathes is one of their clients. And this is not far from the truth. This is a very young and burgeoning science. And this is kind of a window into what is happening in this world in the very early days of the gut-lung axis, understanding therapeutics that affect the lungs that are related to the gut, and various things related to all the major aspects that he's developing in his companies and how to implement this in clinical practice and also in our own lives. So without further ado, this is Vivek Lal with ResBiotic Nutrition and some of his other companies. If you like this podcast, don't forget to like and subscribe below so you never miss an episode. Enjoy. Vivek, it's really nice to meet you. Thanks for coming on the Smarter Not Harder podcast. How are you doing today?
Good, Scott. Great to be here.
So I've heard a lot about you already and about the work that you're doing at your companies, and I find it awesome that there's very few people out there that are kind of dovetailing the therapeutic world from the pharmaceutical side along with the supplement side, and then still doing night shifts, neonatology. How do you balance it all, man? I mean, is it you just are superhuman, or do you have like really good teams? Or
I think you got to have great people sit on the team and learn to delegate, I guess.
Yes, it's always a work in progress. You're marrying a lot of these worlds together, which I think makes it a lot more powerful. So if you're looking at people conventionally or alternatively, like you have a solution and have an ecosystem. And this ecosystem has to do with the gut-lung axis. And this is a newer axis. We know about, obviously, the gut-brain axis, the gut microbiota brain axis. How did you get involved in the study of this, Vivek? And then what is this all about? And how should we think about this clinically from your perspective? Maybe high level first, and then we could get into some of the details. But I'd love to hear your story on how you got involved in this too.
Absolutely. So by background, I'm a double board certified physician, ICU physician, neonatologist. After my clinical training, I got into studying lung biology. So I was at UT Southwestern in the division of Pulmonary and Vascular Biology, learning all these individual molecules, modulating lung inflammation, lung signaling. After doing all of this, realized that people have made their careers on single molecules, but no therapies do come out of it that often. Because lungs are complex, the bodies are complex, right? So how do you just pick one molecule and expect it to be a magic bullet? So that's when I started studying systems biology, omics approaches, right? This is around like 15 years ago. Microbiomics, proteomics, metabolomics. So in those days, the only omics which was a problem was economics, right? Everything was so expensive, right?
Yeah, that's funny. I like that. I haven't heard that before, but that's good. Yeah.
But after doing a lot of omic analysis on the lungs, we started with neonatal babies, right? I'm a neonatologist. So our first hit was chronic lung disease in prematurity, which is very similar to COPD and other chronic lung diseases, but at different spectrum of life.
Interesting. Okay.
So we started studying that in animal models and human samples, and consistently found that a change or a difference in microbial milieu either predisposed a baby to a chronic lung disease or prevented a chronic lung disease, even if you adjusted all the other factors. So that was the aha moment. Oh my God, like what is this, right? We didn't even know what a microbiome was. So when NIH came up with the Human Microbiome Project around, I don't know, it was 2006 or 7, lung was not a target organ. It was like different organs they came up with. We started studying the microbiomics of the lung, found that lungs are not sterile, found that there is constant signal transduction going on between cells of the lung and the microbiome. Although it's not like gut, that is a huge, immune organ with multiple trillions of bacteria. Lung has bacteria, but it's very low biomass. But low biomass doesn't mean that it's not important, right?
Right.
Could be low in number, but could be modulating a lot of things. That's what we found in our clinical studies, that if there was specific kind of bacteria in the lungs, these infants, were born with that specific kind of bacteria, they were more likely to either develop a disease or be resilient from a disease. Right? So that was the first sign of there was a lung microbiome. Then we started studying how does a gut communicate with the lung,
Right.
So that is the gut-lung axis. Essentially, the microbiome of the gut, how does it modulate lung inflammation?
Yeah.
Through the gut-lung axis. But how does the gut communicate with the lungs? It's through the lymphatics, through the blood vessels. So all these bacteria in the gut produce specific metabolites, which enter either the blood vessels or the lymphatics, travel to different parts of the body, including the lungs. That is the gut-lung axis. In addition, every minute, we are breathing and we are eating, we do aspirate something, micro aspirations, especially in babies and kids, more so than adults, but we often aspirate these gut, bacteria. So that also contributes to the lung microbiome. Majorly, the gut-lung axis is modulated by the metabolites which travel between the gut and the lung. And
I see.
Is in the gut through the gut microbiome.
Understood. Okay, so we're talking about metabolites that are being made by gut bacteria. So maybe you can give us maybe just a kind of an overview of some of those that are most important. I mean, we know that there's short-chain fatty acids, of course, that are made by gut bacteria. My guess is that's involved here, but I'd love to hear more about what kinds of, because you're talking about signal transduction, right? And I think that's probably what you're describing here, which is that these are signals coming from the gut via various metabolites that are causing various effects in the lungs.
Absolutely. So small chain fatty acids, you rightly said, is one of the main ones, right? So the specific gut bacteria can increase SCFAs or decrease SCFAs, and increase in SCFA or small chain fatty acid does decrease the inflammation in the lungs. So that is the first finding
Which specific ones? Are there specific short chains that do it more than others, or all?
No, most of these, mostly propionic, and there are some others. But the main mechanism I want to point out is through neutrophilic inflammation. Chronic conditions which happen in the lung are driven by neutrophilic inflammation. Most of them have neutrophilic inflammation as a major component pathogenesis, right? Like COPD, neutrophilic asthma, cystic fibrosis, even COVID has, neutrophilic component. So our labs have been studying neutrophilic inflammation in lungs for a while. What we found was all the extracellular matrix does break down and produce small peptides cleaved by metalloproteinases and produce ACPGP. That is a tripeptide, which increases your neutrophilic inflammation further. So
it's like a cycle, yeah.
Cyclic process. So if you could reduce MMP9 somehow, you could decrease the neutrophilic inflammation. Then we started seeing there were specific bacteria which would downregulate your MMP9 signaling.
Yeah, and are these bacteria that are introduced, so they're in the lungs that are causing the downregulation inflammation. So is it that you have to repopulate the lungs with this bacteria, or is it that you have to repopulate the gut with certain bacteria? Which is the way, or is it both?
Yeah, great question. In the ideal world, I would repopulate the lung. And that's why I started off with a therapeutic company called Alveolus Bio, developing the world's first inhaled microbiota therapy. We call it ResMet, which is a separate, business that is going into clinical trials, hopefully in the next year or so, with inhaled lactobacilli. Because the gut-lung axis is amazing, right? It reduces the inflammation in the lung. But what if you have a COPD or you have IPF, where the inflammation is so severe in the alveolar space that just mere translocation of your metabolites won't work, right? And wouldn't concentrate enough. So you need direct targeting on that area. So that's where inhalation comes into play. But the gut-lung axis does reduce your inflammation in general.
When you're talking about an inhaled therapy here, you're talking about repopulating the lung with certain types of strains of bacteria that you have found with your research and some of the stuff that you published that have shown to decrease some of that inflammatory neutrophilic infiltration.
Yes. So lungs are not like gut that you give a bacteria and they go replicate, right? So lungs, with innate immunity, they just clear it out. So we find that in 32 hours, right, all these bacteria are gone.
Interesting. Yeah. But there's a core population that's still there, but for the most part.
And the core population also, the low biomass, I keep saying, right? It's low biomass. Really little, just because the innate system of the lungs just clear things out. So when we do 16S analysis or when we do microbiome analysis, it detects not only the live bacteria, but also the dead bacteria.
Sure.
Just the signal that this bacteria is, that doesn't mean it's live or dead, right? So it could be just the bacteria.
Of course. Yeah.
But live bacteria are very few in the lungs in general. So when we give live bacteria to decrease the inflammation, they do die, but the metabolites remain, right?
That's what I was going to ask you. So this is similar to what we've thought about with the GI system, right? Like you could even have dead bacteria that are causing a trophic effect or pleiotropic effect in the lungs itself.
That's how these postbiotics work, right? So we have seen, even at ResBiotic, we've launched now a postbiotic line because these things work. And there are no stability issues, just that the doses would be higher than what just, a replicating bacteria would need.
Understood. So like when you're looking at various types of bacteria or strains that have seemed to be most effective here, which of the ones have you found have the most research, or the ones that you found most associated with pulmonary disease and also potentially, respiratory health maybe on the other end of things, right?
Absolutely. So specific lactobacilli, at the phyla level, bacteria, the ones which are beneficial for the lungs are firmicutes. Firmicutes include several like lactobacilli. So specific lactobacilli that we have identified, which we isolated from humans, we have used them as therapy or as supplement, and they seem to decrease this neutrophilic inflammation pretty robustly. And things like proteobacteria, like the gram-negatives, E. coli, Klebsiella, etc., seem to be harmful.
Yeah, I was about to say, like, those are probably not good for the lungs. Okay. Yeah. And so you can find, obviously, people that have, in infants with premature lung disease or premature lung issues because of, I guess, being on a respiratory, et cetera, you're seeing more preponderance of some of these more negatively associated bacterial strains with pulmonary conditions then.
Absolutely. And not just after establishment of disease, even very early, as soon as they're born, if they have these quote unquote good bacteria, they're less likely to develop the chronic disease, even if you expose them to the other, the ventilator injury or oxygen injury. Similarly for adults, right? COPD is caused by cigarette smoke. We all know that. But are there things which potentiate this injury? Or why is that you could smoke a pack a day for 30 years and not get COPD? Why is that I could smoke or not smoke and still get COPD, right?
And still get COPD.
Which we don't know. the things that we don't know. And that's where this microbiome field comes into play. And that's why I'm so excited with this, because it's new, still finding out things every day. And your predisposition or resilience to chronic lung disease depends on your microbial milieu in the gut and lungs.
So what you're telling me, Vivek, is that there could be a strain, I don't even want to say this out loud, but there could be a strain of bacteria that you could, inhale on a daily basis. And like you could potentially like be able to smoke for 30 years and have no problem, compared to, this is a reductionist kind of way to think about it.
It is a reductionist way of thinking about it, but that is absolutely what I'm saying.
Right.
Not that we are proposing.
Of course.
Not propagating bad habits, but yes, your predisposition or your resilience to a disease process, whether it's a lung disease or other disease, is dependent on your microbial milieu or the lungs or the gut, which is very, very under-identified at this time. Science does not know anything at this time, and we are all trying to find it out.
Got it. Yeah, and I think that's so interesting, right? Because we think about predisposition to almost any condition, like you said. Again, there's such a significant microbiome angle here that none of us even knew about when we were in medical school, right?
We saw this at COVID, right? Like, you and I could look perfectly healthy, but I got COVID and I could have been in the ICU and you were just walking around with sniffles, right? So why was that? So was it just the genetic predisposition, or was it your lung microbiome acting up? Was it your gut microbiome? So there are a lot of studies now being done on it, and people are increasingly identifying microbiome to be a major factor in all of these.
Part of the interruption. This episode is brought to you by Health Optimization Medicine and Practice Association, a nonprofit organization training practitioners how to optimize health rather than treat disease. For this episode, check out our gut immune system module. This is how you can learn how to optimize the gut of your clinician. You need to learn this. This is such a big part of clinical practice now. In this particular podcast, we talk about the gut-lung axis. Well, how do you optimize the gut? You actually test it. You look for inflammation, maldigestion, metabolic imbalance, infection, and other aspects, pathogenic bacteria, overgrowth. You can look at all these aspects, optimizing them using probiotics, prebiotics, postbiotics, and other types of modalities as well altogether. So check it out at homehope.org and save 10% on this module or any module of our essential certification by using code podcast10 at checkout. Now back to the show. Did you see some of those studies looking at how smokers fared better during COVID? And they thought this was related to the nicotine itself. Did you see any?
Yeah, which is so strange, right? And people don't talk about these things, but I did see that. I don't know whether I believe it or not, but yes, it is out there, right? So could it be that your smoking was affecting your microbiome in a certain way, which was protective? I don't know. And again, let's not make any public comments about it. But there are things which modulate your microbiome. Smoking, eating some specific things.
Sure.
Protective. So it's an indirect effect also.
Yeah, no, I think that's super interesting. So like one of the things I would wonder in studying lung microbiota is how can you do it easily? Like in the sense that is it easy to sample lung microbiota without contaminating in other locations? Like how do you do that from a practical perspective, and how do you address that in your lab when you're doing testing and stuff?
That's a very good question. It's not easy is a short answer, because anything which is low biomass is more prone to contamination. The ideal way of doing it would be, if I have to check your lung microbiota, I'll do a biopsy into it, right? So I'll do a lung biopsy, or I'll
a bronchoscopy,
yeah, bronchoscopy, and then take a biopsy from there. Or expectorated sputum is what people use as a surrogate, and people have done comparison between upper airway and lower airway with the same patient at the same time, and they're different. Then you have to control for contaminants, based on algorithms and microbial analysis, analytic tools.
Yeah, so this is probably something you had to pioneer as far as how to be able to do this in a...
Yeah, and not just us. We were the first labs to do it, but there are several other labs in Michigan and New York and other places around the world who are increasingly studying this for the last 10 years and have come up with methods. You always have to have a negative control. So whenever you sample, you have to have a negative control in the same. Even the air, right? Yeah,
exactly.
So you have to have an empty bottle with air, and then you sample that and show that, okay, there was nothing there. So
yeah, it's funny. I was just listening to a podcast recently about looking at old extinct animal DNA, and for a long period of time, really what everybody was finding was just the people that were just hanging out and actually getting the DNA from the ground, and it was like their chicken that they're eating for lunch or something like that. So like it seems like a very difficult proposal. So you have to sample the air, and then the gold standard would then be like a bronchoscopy, or would be like a lung biopsy.
Gold standard would do a bronchoscopy and get a deep sample, but
Right.
not usually possible. So you use surrogates such as expectorated sputum in adults. In an infant, you would take a tracheal aspirate. When they're intubated, you take
Right.
a little tracheal aspirate from that. Yeah, people have tried to compare oral microbiota with lung microbiota, assuming that it might be the same, and you'll just use that as a surrogate, but it's not true. It's like different.
Yeah,
different.
it's very. I would imagine it's very different, right, compared to the.
Yeah, even nasopharyngeal is different. Yeah.
Yeah, and so there's low biomass, but it's a very powerful biomass is what it comes down to. And there's a lot of dead biomass there that's also causing a pleiotropic effect.
Yeah, and a lot of metabolites, which we don't study enough, right? Like which are causing the effect. Things like we keep hearing about cystic fibrosis and pseudomonas, right? But why pseudomonas? Why? So there are genetic predispositions to certain microbial milieu too. And we just don't understand it enough. It's just a 10-year-old field,
Yeah, that's what I think it's so interesting. That's why I don't know a lot about it. Obviously, a lot of people don't. That's why I'm having these kind of maybe curious and maybe very basic questions. But when it comes down to like the metabolic activity of the microbiota in the lungs, like when you say Pseudomonas, for example, are you sort of alluding, well, maybe there are certain metabolic products that are being made by the lung microbiota that are causing certain types of bacteria to kind of colonize more than others? Is that what your thought is? Or
it is possible. And, when we talk about microbiome, we just talk about bacteria. But what about the virus? What about the fungi? What about all the other, bacteria, microbial kingdom? We haven't even reached there. So there is a crosstalk going on, right? So we have, X number of cells in the human body, but we have more than 10X of microbial cells.
Yeah.
You've nothing but a bunch of bacteria there,
that's all. Yeah. Yeah. We have like bacteria, fungus, and virus that basically are, they outnumber us vastly. And we call ourselves human, but like we're really.
What a bunch of bacteria, point being, they are talking every second with our human cells and changing it, right? So if you just change the meiosis by just giving extra of something or decreasing something, that would change your human cell activity. Wouldn't you?
Yeah, bacterial interkingdom crosstalk is well established. I think somebody got the Nobel Prize in science for that several years ago. Like there's vesicles that can transfer like information between fungus, virus, archaea, like everything else, right? So like it's crazy what's that.
Produce these exosomes, even bacteria produce these exosomes, and they go to another cell and crosstalk and do the signaling. It's exciting, very exciting.
Yeah, yeah. So from the therapeutic perspective in the lungs, we can start there. So you inhaled probiotic strains is what it sounds like you're doing as far as the therapeutic company. Like, what have you been able to see, at least in like preclinical data? I know you said you're kind of early on in the process still looking to go to trials, but what have you seen happen? What are the things that you've seen change as a result of taking the inhaled side?
Absolutely. So we are done with the preclinical side of at least a couple of disease models, and very potent downregulation of MMP9 signaling, PGP signaling, and neutrophilic inflammation.
Okay.
So I don't say that we are a microbiome company, Alveolus Bio, because it's a bacteria used as a drug here. So irrespective of your microbiome, irrespective of where that neutrophilic inflammation came from, we are claiming that we are downregulating the neutrophilic inflammation, and the only other thing on the market which reduces neutrophilic inflammation is steroids.
Yeah, steroids.
So I know what steroids do, right? To avoid all these inhaled steroids for asthma, for COPD, etc., that people use, in addition to the lava regimen. So my vision there is, how do we use air as medicine? How do we use inhaled lactobacilli to reduce use of steroids or as a complementary adjunct to steroids, right? The downregulation of neutrophilic inflammation is a primary mechanism, but we see reduction in all different kinds of inflammation also, similar to steroids. We have done head-to-head comparison with steroids in all kinds of animal models. We have done smoke animal models. We have done, hyperoxia animal models. We have done pancreatic lipase animal model. Several animal models we have presented to FDA, and they like what they saw and the data.
You said pancreatic lipase model. What model would that be corresponding to?
So it's essentially elastase model, pancreatic elastase model, not lipase model.
Okay.
Elastase essentially breaks down your tissue, lung tissue,
right.
causes injury, right? So
understood.
Elastase model.
Okay, I apologize. I didn't hear that correctly. Has there been any sort of interest in looking at pulmonary infections aside in this case as well? Maybe that's something down the line.
Infections are a huge open Pandora's box, right? Like it's essentially a function of biofilm. All these biofilms are so we have not specifically looked at infections, honestly, but we have some collaborators who want to use our thing for infections.
Yeah, I can imagine this is huge. Just like we're talking about now for the gut, which we'll get to in a minute, but the idea is that you can use certain types of strains of bacteria, in this case, to actually work on an actual gut infection, right? I would imagine there's a lot of interest. We know the sort of the overall burden of pulmonary disease, pulmonary infection, and how often this could become such a huge issue. Like if you could take something more prophylactically, for example, if you have CF, cystic fibrosis, and you're preventing pseudomonal colonization or something like that, that could be huge. It was a huge field, man.
Like huge field, and even starting at the upper airway, chronic sinusitis is probably everyone has it, right? And people don't have a treatment for it. People keep inhaling the steroids and antibiotics.
This stuff always comes back.
So one of our indications is sinusitis. Could be used this for chronic sinusitis. So a wide open field, huge prospect, and we're just getting started there. Yeah.
Yeah, I love it. I can see like a million potential applications for this, just because of working as a hospitalist.
Yeah, I keep joking. One day, air is medicine. All our air conditions could have a probiotic chamber, right? So
I dig it. Right?
If we could inhale the good stuff, we inhale the bad stuff all the time.
Oh, I love it. Like your air conditioner comes with, that's perfect. This is it's like oral microbiome health is like oral microbiome health is like your toothbrush. Like you have your floss is all microbiome optimized, and then your air conditioning, so you're going to have your air purifier with like a canister in there with air is medicine's fun. I love that I'm sure you're using that a lot, and it's great. You're not going as so far to say is that, have you heard of a breatharian before? Do you know what breatharians are? These are people that think that you can live just on air. It's a prana, like the energy from the air. So I don't think you're going that way, Vivek, but I think that what you're saying is that, air is medicine. What's in the air, we can use this and to leverage for not only pulmonary health, but also systemic health, which is... Yeah. And that goes to my next question, I guess, is in related to your study and omics in general. You've kind of thought about the omics, you've gone through all these sort of multiple system aspects. How do you put it all together? In the sense of looking at it from an omics perspective, along with the gut-lung axis.
So just doing omics is a bunch of data we sit on and don't do anything with, right? That's okay. Yeah. You have to know how to integrate these omics. So pathway analysis in a organized fashion is what you have to do. So IPA analysis, you have to look at big picture signals and then validate it at a smaller level. So what omics helps me in is from millions of data points, it gets me to maybe 10 or 15 hits. But those 10 or 15 hits now have to be integrated with each other and then proven at a gene level or a cellular level or RNA level in traditional methods, right? Traditional research methods in gain of function and loss of function experiments. So gain of function and loss of function experiments, what people used to do is they would just pick any of their favorite molecule and they do gain of function, loss of function, show that, okay, this is happening. But what Omics has done is it has just shortlisted that in an unbiased fashion and given us the top hits, and then you take that to the traditional method. That's how I use Omics.
And just to be clear, gain of function has also gotten a little bit of a bad rap since COVID. So I'm making sure you're not doing anything crazy. You're not doing anything crazy in your lab, right, Vivek? We're not talking about nothing like the new COVID coming in. Right? We're good.
That was a shortcut to being famous there, but yeah, no.
Infamous, maybe.
Infamous, yes.
Yeah. So you're creating these models. Are you using has artificial intelligence come into the model or into manipulate it for you?
Absolutely. So when you get all this data, you use AI, train them, and then just essentially come up with your top molecules based on that, right? In the future, I think in the drug development space, in FDA modernization, people might move away from all these animal research, etc., and then do organoids and cellular models with use of AI and unbiased omic analysis.
Organoids. So you're talking about like just an artificial lung, or not artificial, like a human lung.
We use it a lot. So we have an end-to-end facility, right, with using cells, which is 2D cells or 3D organoids. Yeah. We grow lungs essentially as organoids and then test, specific bacteria on it or a group of bacteria on it. And then we go to the animal models, you check. So we had all of these models and infrastructure before going to clinical trials. And that's when I thought, oh my God, this supplement industry is a bunch of snake oil. Why doesn't someone try to scientify the supplement industry, right? Why don't they do drug development level research in supplement industry? And after doing some diligence, understood that it's a cost thing. Putting the infrastructure for all these preclinical work and then going to the clinical trials is extremely expensive and they don't get the ROI. So that's why people don't do it. They just take the shortcut.
Yep.
I realized I already have the infrastructure while doing the drug development company, right? So why not use it in a cost-effective way and try to create supplements which are like drugs, but not drugs, The
rigor's there, and then you'd also have the sort of the pharma quality to the products as well. So you're using the same strains that you're developing on the pharmaceutical side, but you're just using them now in just a different route of delivery, talking about the gut. Is that the major difference, or is there any...
There are some other differences. So we do have a strain bank that we have created. For pharmaceuticals side, we are calling them LBP, Live Biotherapeutics product, right? So the production quality is similar, but they have to go through several other checks.
Okay.
For the ones which would be inhaled. And FDA has those requirements. But in our case, we are kind of doing the same thing for supplement side. Just that it's like two ends of the spectrum. Drug is so difficult through an FDA. Supplement is so easy. I always suppose there needs to be a middle ground for...
Yeah. Actually, I love that you're mentioning that because actually something that's when we've been trying to develop at our company. It's called Transcriptions, and we make pharmaceutical grade products in the form of a buccal troche. Yeah. And we always complain about the same thing, which is that you have your pharma, and then you have your supplement companies, which have no regulations at all. They can self-regulate, and some companies do. We do. We get all of our certificates of analysis done third party with, different third parties and the manufacturers gave us for the ingredients. We're doing all of our batch testing. We're doing all of the microbial, doing everything every single time. And there are a couple of supplement aggregators out there that I know, that Karen knows and that you probably know as well, that are doing more work to try to do the legwork of what compounds are on their platforms. But you're right. In general, it's very easy. And I do think there needs to be a middle ground. And so maybe we can figure out what we need to call that, Vivek, because that we play around with it. For us, we have, internally we call it fun pharma, where we're having fun. We have products that can really help people, but we're also pharmaceutical grade. So maybe if you like that, we can talk about it.
Yeah. Let's talk about that.
Part of the interruption. This episode is brought to you by Transcriptions, makers of precision dosed, physician formulated, and pharmaceutical grade formulas in the form of a buccal troche. For this episode, check out our Tromune. Tromune is high potency cordyceps extract cordycepin. It is about 100 times more potent as an anti-inflammatory, as an immune system modulator, as an antiviral, and it also probably optimizes gut health as well, shifting their biology, shifting microbiota into a more optimized state of health. So check it out at transcriptions.com and save 10% on your order by using code POD10 at checkout. Now back to the show.
I refer to ResBiotic Nutrition as a biotech CPG company, essentially, right? All our products are patented. Our ingredients are proprietary, and we're doing the same amount of rigor from the preclinical perspective and then the clinical trial. So might as well create a new category on that.
Yeah, let's do it. But so from a gut perspective, how do you think it's working to give some of these strains that you've studied? Like, how's it working to give it in the gut, make sure it gets to the right place, and then how is it causing this sort of more pleiotropic effect in the lungs from there, as far as you can tell?
So it's, mainly because of the metabolites they're creating and overwhelming the system with the beneficial bacteria so that you overwhelm the system with the beneficial metabolites, which not only go to the lungs, but go to the other things or other organs also, right? But if they are useful in the lungs, they would decrease the inflammation in the lungs and improve breathing. So for our lung product, ResBiotic Lung Support, we had to create a category. No one knew that probiotics could work for the gut-lung axis or lung. So we had to essentially create a category because we were seeing benefits in people who had chronic lung issues, right? Diseases, we can't make disease claims with supplements, but a lot of our customers would call us that, hey, I have this and now I'm, doing better. My pulmonologist doesn't believe it, right? So that was
right.
And the second category was people who were just exposed to bad air, whether it is pollution, wildfires, or smoking. I did not know smoking was a category. And then people started saying that, hey, every single time I've met a doctor, they've just, ridiculed me and told me to stop quitting. I have made a decision not to quit. What can I take to reduce my inflammation, right? So those kind of comments started coming in, and smokers or people who are trying to quit became a category. And then athletes who wanted to improve their FEV1 or just vital capacity became a category.
Have you seen some benefit there on the endurance athletic side?
Endurance and swimmers, and again, these are all anecdotal experiences.
Sure.
Feedback testimonials. I've not done an RCT on athletes.
I understand. Yeah.
We have done our RCTs, like we have done 2 or 3 clinical trials already on ResBiotic support, but you can't do it for every indication. But these are the testimonials we get, and so our running joke became that anyone who breathes is a customer.
I love that. Yeah, I mean, look, that's probably not that far off. But so the clinical trials that you have done so far using it, tell me a little bit about the conditions or indications and study results that you've seen.
So we used it in healthy and smokers and in asthmatics. And we saw, again, I have to be careful in what I say here, but we saw benefits in pulmonary functions. We saw benefit in inflammatory markers. We saw benefit in microbiota. Similarly, we did a double-blind RCT on COPD patients and, healthy placebo control, and found benefits there in quality of life, in sleep, in various markers, biomarkers. So we were very happy.
Yeah, I can imagine. And so, question for me is, I know that microbiota can change very quickly within a couple hours, a couple days at the least. It can happen very quickly. So what are you seeing as far as how long it takes once you start inoculating, the gut with these strains, or inoculating the lungs? Maybe it's a different time course, but what are you looking at? We're looking at days, weeks, months, years. What's the time course for people to see a benefit typically?
Yeah. So these are probiotics which show gut benefits within a week, like better digestion, you're pooping well, those kind of things within a week. But the lung inflammation, bringing down the inflammation and lung benefits do take time. I've seen reports within a week, which I don't believe, but people call us, hey, I'm better in a week, right?
Yeah.
Usually, most of the feedback is in the 3 to 4 week period. Our clinical trials were done for, a month and 2 months. So 3 to 4 week period. So we recommend people be on it for at least 3 months. But, most of our customers don't leave it because they start seeing the benefit and they just continue on it. Like my family has been on it for 4 years now, right? So
sure.
Even have any lung issues.
Yeah. I was going to ask you about engraftment. I guess maybe that's part of it. Which is, do you think what's happening here in the lungs or in the gut is caused because the bacteria, and maybe actually you can just delineate which ones you're actually using. You said the lactobacillus strains, but
using specific lactobacilli strains, one plantarum, one acidophilus, and one rhamnosus. These are RSB strains, which are specific proprietary.
Right, these proprietary strains. And so do you see it as an engrafting, or do you see it more of this is something that causes the pleiotropic effect, and then as a result, you have the change?
Yeah, I think it is pleiotropic effect, reducing or increasing metabolites, which are anti-inflammatory. And so I've not encountered patients who come and say that, hey, I've stopped it for 6 months and check me again. So we have not dealt with that.
Yeah, got it. Yeah.
So the people just continue it, because they're seeing the benefit, they don't want to quit.
Would there be any detriment to, you think, continue it, in the sense that, and sometimes we have different types of bacteria that, like for example, it's really a good idea to cycle things like spores, like Probiotics. We talk about from a gut perspective, Bacillus species tend to be better, 1 month to 3 months on, and then, 3 months off. Do you have any sense of that at this point that would be required or helpful?
I think it's because these are lactobacilli, which have been well established to be very safe and are in the we extracted these from humans, right? So they are part of our natural immune system. And so, no, we have not had any issues.
Okay. So there's no concern either from
issues of bacterial overgrowth or anything. Yeah, we have not seen anything.
Okay. I mean, yeah, you're right. I mean, Lactobacillus bifidobacterium are your two most common species. I know often times lactobacillus has also been associated with the vagus nerve and GABAergic transmission and signaling. Have you seen any of that? And how that might relate to the lungs, or I know it relates to the brain, but I don't know if there's any lung.
It does relate to the brain because the metabolites which are produced in the gut does go to the brain also, right? Vagus or just to the blood or lymphatics, it does go to the brain. And again, anecdotal and customer feedback. They sleep better, they feel better. I know at least a few people with, rheumatic issues or major inflammatory issues who have gotten back to us that they are on ResBiotic lung support for that. So our clinical trials were done on lung issues. That's why we don't claim anything else, but it doesn't mean that other things, are not helped.
Yeah, I mean, I'm just thinking about in combination. We use something called methylene blue at our company, Transcriptions, which is it's a compound been around a long time. You probably have heard of it from like a rescue perspective in septic shock and things like that. Maybe in the NICU they use it in babies. I don't know, but there's also some indication that it helps with hypoxic stress. And so when people come to altitude here in Colorado, where I live, I'll often give them some methylene blue before they get on an airplane because it helps with that hypoxic stress. And you have a lot of people in my neck of the woods that are here in Colorado training high so that they can go and, compete back at sea level. So I wonder if, some of these teams, if you're saying like this is increasing FEV1, it'd be a nice and interesting combination. Yeah,
absolutely. It might be, and there's so many things like that, which people have just, us physicians have, kind of written off, right? All these years claiming science. I feel this science just was not done enough. It doesn't mean that it doesn't work, right? These kind of things, you have to do the science on it to prove that it works. Just that you've not done it doesn't mean that it doesn't work.
Exactly. Yeah. And it's hard, right? As you mentioned, most of these companies, like supplement companies, do not have the funds to be able to fund a study on a randomized clinical trial on COPD and a gut bacteria strain to use. It's a novel position to be in to be able to do that.
It is. And it's not that they don't have the funds, but if they spend $1 million on marketing, they will make $5 million out of it. But if they spend $1 million on a clinical trial, you have to start from zero again to spend that $1 million for marketing. So that is the catch, right? People look for the short-term result.
Yeah. How did you end up convincing your investors to do it? Like, was it another sort of route? Because it's hard. I guess I'm sort of selfishly asking this because, as a company, we would love to do more studies, but we don't do them, at least yet, because we don't have the funding to be able to do it. Right. So did you create like a whole system where everything's getting funded through the same verticals, or do you have different funding depending on the situation for your projects?
So we have a CRO of our own called Able Biolabs,
Okay.
So we subsidize all our studies there, clinical trials, etc. And we can do it for a fraction for us, and we don't advertise it. It's kind of a boutique for us and friends and family. So we do it for lower cost than anywhere else you'll find. But it's still some cost, and that you have to show the vision to the investors to convince them that this is the future. Because I firmly believe this is the future even from the supplement industry. Like in 5 years from now, people will get educated. They're already getting educated about the science and what you should take and what you should not take inside your body, right? So people are getting educated. All these legacy marketing snake oil brands would wean out and wean out or adopt these newer science-based formulas.
Yeah. Well, this is super cool, Vivek. I mean, I think it's really exciting for me too, because on October 17th and 18th here in Boulder, Colorado, you're going to be speaking at our symposium. It's called the Health Optimization Medicine and Practices Symposium. So I'm guessing you're going to talk about something related to these topics, right?
I will, hopefully. Yeah. We can talk about other things also, but yeah, I think we'll stick to something around these.
Yeah, so I would love you to talk about, this intersection of the gut-lung axis and sort of the multi-omics approach, and the whole conference is really about what's on the forefront of medicine, but from a health perspective, which is unique most of the time, and it's super important what you're doing with, with COPD and advanced lung disease, but if we can keep people healthy, like you're taking your product, I take my products all the time to, optimize health, like it's so much easier to address something that might go wrong if your system's in a better place ahead of time.
Absolutely. We have a rule in our company, nothing goes out which we can't consume or we won't take, right? So we are the first guinea pig for all our clinical trials. And that's the way to do it, right?
I'm 100% with you. And we always say that at our nonprofit as well, is that we wouldn't give you a supplement that we wouldn't have tried ourselves. Now, there might be some exceptions. So if I'm male, I may not give myself estrogen, for example. Or if I'm female, it would be like high doses of testosterone. But so there are exceptions. But in general, yes, if, you got to walk the talk, and walk the walk, right? And so I think it's great. So have you been to Boulder, Colorado before?
I have, but now I have a better reason to go come.
Yeah, so it's going to be fun, October 17th and 18th. Hope some of you that are listening will join us. Vivek is going to talk all about the gut-lung axis and much more, and hopefully give us some samples to try. I'm sure you will, or I'll make Kara bring some with her if she can from Colorado. But at the end of the podcast, Vivek, I also ask everybody like the same question, which is, the name of the podcast is called the Smarter Not Harder Podcast. And the reason why we call it that is that, we try to live smarter, not harder. And we've talked about various ways that you think about this already, probably from a gut-lung axis perspective, but in all sort of walks of life and all the aspects of your experience up until now, neonatology, dad, husband, whatever else you want to say, what are 3 ways that you feel people can live smarter, not harder?
I think the first way is they have to take control of their own health. There is a lot out there, whether from your peers, whether from the internet, whether from, legacy health advisors, right? So you have to educate yourself. I think unless people start educating themselves, no one can help that. So that is the first thing. Second, I think incorporating healthy habits in life as early as possible in life. We all spend our teens and 20s and 30s, not caring about the 40s and 50s and 60s, but I feel that it doesn't matter. So educating the new generation that earlier you take control of your health, the better it is, right? And I think third is what we eat and what we breathe is important.
Yes, I love it. Those are simple yet super powerful, man. So thank you. So for those that are listening, please let them know where they can find out more about you, more about your companies, the products that you have in brief, websites, anything else you'd like to share.
Absolutely. So ResBiotic Nutrition is our biotech CPG company.
I love that.
Hands back supplements. ResBiotic, ResBiotic.com. ResBiotic stands for Restoration Biotic. We make supplements to restore health from chronic issues or to prevent chronic issues. You can, find me there, or my email would be info@resbiotic.com.
I love that your customer base is anybody that breathes. That's good. I dig it.
Anybody that breathes is the right customer base. Yes.
Well, cool, man. Well, I'm really excited to meet you in person in October. Thanks so much for spending some time with me today.
Wonderful, Scott. Likewise. Wonderful talking to you today.
Thanks so much for tuning in to another episode of the Smarter Not Harder podcast. This is a fantastic episode. I learned so much about the gut-lung axis, the multi-omics approach that it took to get there, and Vivek's companies and how he's doing various things from an aeration perspective and from a gut perspective to optimize pulmonary health. This is such a big deal and so many potential implications. You guys are hearing it first, probably. This is the first I've learned about this. This is a new field that hasn't been around that long, and there's so much more to come. So check out Vivek at our symposium coming up October 17th and 18th in Boulder, Colorado. Check it out at homehope.org. If you like this podcast, don't forget to like and subscribe below so you never miss an episode. Share with your friends and family, and we'll check you next time on the Smarter Not Harder podcast. Take care.